So today, in the first talk, we're very happy to have Pierre Dechant. He's going to tell us about virus, polytope, and machine learning. Let's welcome Pierre.
Thank you very much for the kind introduction and also for the invitation for me to speak here. Thanks to all the organizers for putting on such a great meeting again. I've got very fond memories of the symposium 4 years ago. I think I was talking about rather more algebraic things back then, but with the last 18 months that we've had, I thought I couldn't not talk about viruses. So at the beginning, I'll give you a whistle-stop tour about virus structure and how that's related to algebraic and geometric ideas. Then once you understand virus structure, you can also wonder how viruses assemble these structures. So the last 10 years have seen a revolution in our understanding of how certain types of viruses assemble through something called packaging signal mediated assembly. And the story I really want to tell you today is about how we can use these insights into virus structure and assembly to start messing with this as an antiviral approach. So a paper that's just come out brand new is about how we can use these insights in something called therapeutic interfering particles. So that's trying to construct some competitor for that parasitizes the viruses themselves and tries to derail them in their infection. So beating viruses at their own game. So stay with me till then. At the end, if I've got time, I will talk a bit about some machine learning stuff I've done with Yang. If I've got time, I'll talk about that. So it will be a bit of a whistle-stop tour about virus structure. So there's more detail if you're interested in the review article that Ryan Tvarok and I've written this year. There's more detail in the article with Yang on the machine learning stuff, and the brand new paper that I was talking about on therapeutic interfering particles is just out this week. So you all know in the pandemic what a virus is, so I'll just say that it's a piece of genetic information, and because this genetic information is inherently quite fragile, it's normally protected by something called a virus capsid. So that's a protein shell, and at least one of the genes in this genomic material codes for this code protein or capsid protein and makes these shapes that you're all familiar with now. So also the other thing just to clarify is that a lot of viruses can be single-stranded, and single-stranded nucleic acid is quite different from the double-stranded helix that a lot of you will be familiar with, and it can flop around. It's very flexible. It can form lots of transient shapes. It can sort of base pair with itself and form these secondary structures. Excuse me. So it's quite different from the double-stranded helix that is very stable, that is so iconic. So this geometric building block, the capsid coprotein, is essentially just defined by its shape. A lot of proteins have other functions as well, but for virus structure, we just want a building block in a particular shape to make this assembly work. So in early electron micrographs from the 50s, people could see that viruses looked either sort of rod-like or spherical. Here the resolution is a little bit better, but back then they just looked spherical or rod-like, maybe with some granularity, and people could maybe start to see a little bit of structure. But an ingenious argument by Watson and Crick of DNA fame actually deduced in a very nice Nature paper that clearly, because everything was built from discrete building blocks in biology, these particles couldn't be exact spheres or sort of exact cylinders. They had to be helical or polyhedral. And in fact, icosahedral would make the most sense because that's the largest symmetry group available in 3 dimensions. So they hypothesized that these spheres were actually probably icosahedral. And there's of course many different polytopes that you can have that are icosahedral. So the triangular icosahedron is one example, but you can have others with pentagons and rhombuses and kites and so on. So a priori, that's a possibility, and we'll see these guys later. But the icosahedron is also distinguished with these triangles. So we'll zone in on that for a little bit. So an icosahedron consists of 20 triangles, and you could assemble them one by one. And essentially the order in which you assemble that would give you a Hamiltonian path on the dual polytope, which is the dodecahedron. Or you can think of an icosahedral net, which is sort of a fragment of a triangular lattice, and just cut that out and glue together the right edges, and it'll fold up into an icosahedron. So this idea of Hamiltonian paths will sort of underpin various things that I talk about throughout the talk. So that's just from a mathematical perspective, and there's of course many Hamiltonian paths and orders that you could assemble an icosahedron in. But if you're building this with some actual biophysical building blocks, there's also some thermodynamics that comes in. Some pathways that form more bonds early on are going to be thermodynamically favored and assemble exponentially more rapidly. So that's also a consideration that comes in. So the more bonds you can form early on in the process, the better. And then later on, the process is driven to completion, because the more complete the capsid is, the more bonds you're making at each step, and the more thermodynamically favored that is. So you might say that the original problem was for viruses to be able to encode triangles, and if they can make triangles, they can make an icosahedron, and then they're in business. So that's kind of how we'd understood the original biological question in a mathematical way. But by the time that viruses have a building block that is effectively a triangle or a hexagon, then we don't just need to make 20 copies of it. We can make many. Like a gene is a recipe, so you can make many, many copies. You don't need to limit yourself at 20. So actually viruses have evolved to use these triangles a lot more smartly than just using them to make one icosahedron. There's a lot of substructure. So the icosahedral faces are all sort of clad out with some hexagonal looking lattice. And that's because if you've got a triangle and you have this icosahedron made of triangles, you could imagine subdividing each of these large triangles in terms of smaller triangles. So say you make a large triangle out of 4 smaller ones, like here, or 9, and then biologically that makes a lot of sense, because then you're just using the same building block more, and you can make a larger capsid for encoding a smaller gene and a smaller geometric building block. So the icosahedron is distinguished because you can triangulate triangles. You can decompose that in terms of smaller triangles. Now, they're not a symmetry equivalent anymore. They're not exactly icosahedrally equivalent. They lie on different icosahedral orbits. But in terms of the biology of, oh, this is a triangle, it sits in a lattice of triangles, it does very well. So this is sort of a fudge or a compromise, which is called quasi-equivalence, because it's not exactly mathematically equivalent. So this example is a triangulation where each large triangle of the icosahedron has been replaced by 4 small triangles, and that's more or less what hepatitis B virus does. It's only got one structural gene, one building block, and it does this T4 triangulation. So the T number, triangulation number, just counts how many small triangles make up a big triangle. And it turns out there's a very systematic way of doing things. I'll skip over the details slightly, but effectively, if you start with a triangular lattice or a hexagonal lattice, you can sort of put an icosahedral net on that, subject to some symmetry requirements, and then your large icosahedron is going to inherit some of that hexagonal chicken wire pattern from the underlying lattice. And the allowed triangulation numbers that you get are parameterized in terms of 2 integers. So they just count the number of integer steps you go along the 2 different lattice directions from one corner of a triangle to the other corner of the triangle. And that gives certain allowed numbers. Not every T number is allowed in that sense, but you can have a sort of discrete quantized spectrum of allowed geometries. So in general, viruses that have different T numbers have completely different building blocks. So it's not like they were all using the same underlying lattice, but if you could make cages with different triangulation numbers out of the same building blocks, because the triangulation number effectively counts an area, its square root is essentially a length scale, and it should scale with the particle diameter. So in general, as I said, that's not true, but we will see 2 counterexamples coming up. So you can do this fairly systematically. Here's just another picture of a triangular lattice on an icosahedral net on the underlying hexagonal lattice, and you can fold this up into these nano cages. Here it's in terms of a triangulation, but if you go to the dual tiling, you get a pentagon-hexagon tiling immediately. So those two viewpoints are dual. The triangulations are, I guess, more the historical way. Sometimes the pentagons, hexagons are more like what the actual capsid ends up looking like. And also you're probably all familiar with carbon fullerenes that look like these cages. And there's also pentagon, hexagon tiling that is a bit more natural, but the two tilings are just dual to each other. And this is actually Herpes simplex virus. Its T number is 16, and it's one of the more complex viruses. So the simpler ones have 3, 4, 7. So 16 is already quite a complex virus, and T16 would have been thought of as quite a large T number back in the day. But there's a bit of a novel development called giant viruses, and a bit of an embarrassment to virology, because for about a century, people thought by definition viruses would be small, and they didn't really look for large viruses. So once people started looking for large viruses around 15 years ago, they found them everywhere, all sorts of different families, different triangulation numbers, but they all seem to have something in common. They're all quite large, and they've turned these large triangulation numbers into a bit of an art form. So all these little nuggets here are all hexagons, and they've all got this large hexagonal pattern here. So these large triangles, and then called trisymmetrons, and then the pentagons, which are called pentasymmetrons. An alternative view of looking at it is decomposing these pentagons here into 5 little triangles and attaching them to the edges of the large triangle. So you essentially just have 20 large triangles with little hooks on the end. And you get various different families of viruses that have different sizes, but follow the same design pattern of having little hooks and assembling. So there's a lampshade that has a similar design principle. It's just a rhombic tricontrahedron, so made out of sort of rhombus-shaped materials, but these little catches allow you to assemble that manually, and it sticks together on its own. So it seems like these giant viruses all have the same underlying design scheme, and they have a certain pattern where they all have the same H number of 7, which makes this little hook. And then they can explore different Ks, so 18 and 10 and 12 and 8, and various families have different Ks that give you these different ginormous triangulations. But if you think of these 20 large triangles with catches assembling, you're back to the idea of this Hamiltonian path of an icosahedron assembling the 20 faces. If you think of the tiny little triangles as something else, you're back to a Hamiltonian path with a slightly more complicated polytope. So these giant viruses are found everywhere with different triangulation numbers. They seem to be everywhere. And I was just thinking, what if they're all evolutionarily related and they all actually share more or less the same geometric building block? Then their particle diameter should scale with the square root of the T number. And in fact, that seems to be the case quite well. So if you know a particle has a certain size, but you don't know its geometry, you can maybe predict the triangulation number and the geometry of the surface organization of the virus by just using these geometric grounds. So the blue ones are the known examples, and the red ones would just be the family that you get by substituting different integer k's into the triangulation formula and this best line fit. So it seems like all these giant viruses could be evolutionarily related, and they share essentially the same geometric building block, because what's difficult is to evolve a geometric shape that works, and then evolving away from that is going to be very difficult again. So maybe there's evolutionary pressure for them to share that same building block. So I've hinted at Fullerenes earlier, so this idea of Caspar-Klug triangulations carries over exactly more or less to Fullerenes. So you get these cages. Sorry, was there a question?
Oh, yeah, hi, Pierre. Just a quick question, just to get understanding here. So the typical size of the number of triangulation of those giant viruses are
sorry,
they are classified by T number, right?
Yeah, so I said anything up to 16, 25 used to be quite large, but these are now in the hundreds. So the sort of between 147 and 499 is the largest that is actually known. There's one that's around 1,000 where we don't even know exactly what triangulation number it is. It's in a range.
I see.
So they're huge.
So those are, how should I put it? Are they considered as some kind of approximation to like a real giant virus in nature, like in the human bodies and so on?
Not in human bodies, but these guys tend to infect amoeba and algae. So nothing in the human body as far as we know. But yeah, these guys exist in nature. They've been found 15 years ago or over the course of the last 15 years.
I see. So, and I should think about like this, order of 100 kind of triangulation as some good approximation to those giant virus. Is that the point here?
Yeah, exactly. So yeah, this giant one, Cafeteria rhambagensis virus is the largest known triangulation with a triangulation number 499.
I see. So how do we know these are the kind of, I guess my question is really, how are those, how do we know those are the optimal kind of triangulation for those to approximate those virus, or is it just set by the revolution of some electron tunneling telescope?
Yeah, so there's not that many options to make it work, right? So there are discrete allowed numbers, and it could either be 470 or 499 or 550, but not a lot in between, sort of thing. So you can say, oh, it's got to be the 499 one.
Oh, I see.
But yeah, it's
with
just
respect to
looking
those
at
symmetries. I see. Okay.
Yeah, so people do lots of particle averaging over lots of particles in cryo-electron microscopy, and you can you can see the sort of nugget structure emerging, and then they try to fit the best triangulation number that has the best fit to the experimental results.
Oh, that's interesting. Okay, thank you. Yeah, that's my question. Thanks.
Sorry. Yeah, great. Thank you. Yeah, I should have said how huge these guys are. I've got a slide with them all on later, but I've eliminated that for now. So Fullerenes follow exactly the same sort of type of design principles, and because they all use the same carbon bond length, you would expect them to follow the same sort of scaling relation as well. So these Fullerenes have similar versions. They are really the pentagon-hexagon tiling, where each of the spheres is a carbon atom, and the guy down here is essentially just a football, a truncated icosahedron. You can do much the same thing, starting with the dodecahedron, and the next sort of sizes up, insert hexagons in slightly different orientations between the pentagons, and you can just keep going and going, making these larger cages. So going back to the earlier ideas of actually different tile shapes might also work. Turns out that nature, like in physics, everything that isn't forbidden by some fundamental law of nature or mathematical result will actually happen. So I'll just skip through a few examples of where you have different tile shapes. So here's a rhombus tiling virus called MS2, where it's not triangular building blocks, but it's rhombuses. So here might be a triangulation example. Here's a rhombus. Here's a more kite-shaped one. And even though the surface organization looks quite similar across them, actually understanding the tiles tells you a bit more about the assembly, whether it's 2 proteins that are bound together or 3, and how these capsids assemble, and so on. But in these examples, you still have the same tile shape. So everything's made of identical building blocks. But we've even found examples where you need to allow several types of different building blocks like kites and rhombuses to explain what's really going on. And Wright and Sharrock has revolutionized structural virology again, and these mathematical symmetry models of viruses after the early work in the 50s, 60s, where everything was then about triangulations, and that was very successful. But the results, experimental results from the last 10, 20 years have given us such a precision that we now seem to find lots of mismatches where triangulations actually don't work all that well. And we need to make mathematical generalizations to account for that. So a lot of you will be familiar with geodesic domes in architecture, which are essentially these triangulations in architecture. But you could also use these other design principles with, say, kites and rhombuses. And there's a design concept for the Amazon headquarters in Seattle, which is more like a kite rhombus tiling. You can also use these insights into how viruses assemble to construct sort of nanoparticles from scratch, following similar design principles. So here there's some work on self-assembling protein nanoparticles that follow some of the same microscopic rules, like the like forming 3-fold axes or 5-fold axes, and form these polytopes spontaneously according to these local rules of the white guy likes having 5 neighbors, the black guy here likes having 3 neighbors, and so on. So you can understand these mathematical polytopes that are possibilities and classify them as well. And these particles are used in the lab, and they're in fact used for malaria vaccine design. So just to say, you can also decorate the hexagonal lattice a bit more and put an icosahedral net on that and get other more complicated icosahedral tilings that way. And they are actually a better description of the herpes simplex virus that I mentioned earlier. There were sort of some triangular gaps in there that we glossed over, and the hexamers are in slightly different orientations, so allowing these more general Archimedean tilings are necessary for the current experimental resolution that we have. So this was all about describing virus surface architectures. It was all at the sort of spherical level, but the question is, can we somehow also have an imprint of the symmetry from the capsid outside within the virus particle, and why might that be, and why would that make sense? So we were thinking about some affine symmetry ideas. I'll just quickly go over this because obviously for icosahedral symmetry, you can't have an affine extension in the traditional way, but you can sort of construct fragments that are extended and nested structures that are similar to what you might do in order to construct a hexagonal lattice using a translation and then taking the group orbit. You could do the same thing for a non-crystallographic symmetry, translate, take the orbit, and you get these nested structures. So the question is, are they going to be any use for modeling real things like these carbon compounds or viruses? So this is for toy example in 2D, but if you do that for 3D, you get some affine extensions and some translations that effectively construct these larger cages, C180, C320, and so on, the whole family, from the first one, the C80. And these carbon cages, they're like Russian dolls. They can live inside each other. So they can form nested arrangements called carbon onions. And here's another example. When you start with the C60, using the affine translation, you derive the C240 from that, and then the 540 from that. And they follow typical Caspar-Klug type layouts, but they can live one inside each other. So for carbon, Fullerenes affine symmetry ideas are already quite interesting. The question is, do they also make any sense when you try to apply them to viruses? So here we have a cross-section of a virus, and we're trying to model that with some of these point sets that we've got from the affine extensions, some nested structures. And it turns out some viruses have an icosahedrally ordered component of the RNA inside, and that's quite surprising because we always thought that it would be disordered. It would just be shoved in there any old way. And why there is a remnant of icosahedral symmetry in there was a bit of a mystery. And you see this beautiful dodecahedral cage of RNA sitting inside this hemisphere of the capsid. And what's really happening is that this RNA inside forms these secondary structures called stem loops that we saw earlier, and they can bind to capsid protein at the inner capsid surface. And that happens in some symmetry equivalent positions. So you get some imprint of the symmetry on the capsid surface that extends sort of into the particle interior. And that's because seems to be a fundamental assembly process principle, that these RNA stem loops sort of cooperatively work in assembling the capsid around them. So there are specific interactions between RNA and code protein, and that allows these viruses to assemble in the first place. Sometimes the geometric building block only has the right shape once it's bound to the RNA. So it's a really clever way for ensuring that only viral RNA gets packaged rather than just any old cellular RNA that might be floating around. Excuse me. So it's a bit like a smart washing line that could get the clothes itself, or this little octopus that uses all its tentacles to go and fetch these different hats. So the idea is that the virus has some genetic sequence, but that these RNA secondary structures allow that to fold up into these stem loops. These stem loops at the tips can bind code protein, and how well they bind and how well they facilitate assembly determines the fitness of the virus in terms of the assembly efficiency. So you can think of a dodecahedral toy model with 12 pentagons forming a dodecahedron, and you have 12 packaging signals that each sort of bind one of these 12 pentagons, and then the pentagons bind together when they're in proximity to each other, once they've bound these stem loops, and the capsid is assembled around the RNA in that way. So this was all centered around symmetry ideas. What we're going into now is a bit more bioinformatics, computational simulations and modeling. It's still following on in exactly the same vein. So you have this genetic sequence and it codes for some genes and like polymerase and the code protein, but it also has a secondary function, which is this assembly manual. So this genomic sequence codes for genes and proteins, but it also has a secondary structure landscape that allows the virus to assemble efficiently. And these are called packaging signals, and they all sort of fetch a building block and then assemble the viral capsid. So what viruses struggle with is that they've sort of got two orthogonal sets of constraints. One is that they need to code for the right genes. The other one is that they need to have these right secondary structures to assemble efficiently. And this seems to be a reasonably general thing across many viruses. The specifics are different. They might have different kind of stem loops and recognition motifs where they bind code protein, but it's sort of the same idea that they have dispersed packaging signals throughout the genome. So the whole genome is important for genes as well as for assembly. So what you can do with this insight is you can actually do better than the virus. Say the virus needs to sort of find a compromise between two different sets of constraints, genes and the assembly. So maybe their packaging signals aren't ideal, but if you constructed an artificial sequence, doesn't need to code for any genes, you can just focus on optimizing these packaging signals. You can then come up with something that assembles better than the virus itself. And you could use these virus-like particles for similar purposes as these nanoparticles that we'd had earlier, for vaccines, for drug delivery, and so on. So this also opens up a new antiviral approach or range of approaches. If you understand that this RNA code protein contact is crucial for viral assembly, you can start messing with that as a sort of antiviral therapy. So you could find drugs that bind to the RNA sequence at these stem loops instead, so that the code protein can't bind, or you can try to block the code protein so it can't bind to the RNA. That might be quite a good approach, because this hardwired geometric thing is something that the virus can't evolve away from very easily, right? Either it's geometric and it all fits together, or it doesn't. It can't fit together a little bit. So these are the hard rules where geometry really comes into biology. So this might be something useful where viruses can't just escape via evolution. But it is tricky because these are really large numbers we're talking about. Viruses, sort of, as you know, grow exponentially. There's a lot of particles. Even within the particles, there's a lot of these RNA stem loops, a lot of code proteins. So it seems like that's a losing battle, right? There's a lot that you need to get under control lots and lots of particles and these contacts that you would have to inhibit. So the viruses obviously grow exponentially. So the question is, can we do something smarter than this? Can we somehow harness this exponential growth and turn it into an advantage for us so our antivirus strategy also grows exponentially along with the virus? And a lot of you will have heard a lot about PCR recently. It's a gold standard test for viral diagnosis. So a lot of you will have had a COVID PCR test recently. So I worked with them 25 years ago, and it blew my mind how genius this idea is. You start with a microscopic amount of DNA or RNA, and then you just go through different cycles of duplicating what you've got. So you might do 50, 60 cycles, and you start with a microscopic amount, and you end up with a macroscopic amount that you can definitely detect, say, COVID RNA. And it's called the polymerase chain reaction. And what's really powerful about this is this exponential growth, like viruses themselves grow exponentially. So I thought, well, can we harness something like this, this exponential growth? So just to recap, viruses have these sequences that have both the genetic code and the assembly instructions. So they've got this dual compromise and a bit of a dilemma. So the question is, can we design a sequence that is a better assembler than the real virus, doesn't code for any genes, but will be copied by the virus and essentially competes with it for resources. So the virus is busy making polymerase, making code protein, and this therapeutic interfering particle goes and steals it all. So it steals the polymerase that the virus has just created, steals the code proteins that the virus has just created, and sort of drains it of the resources. So it's parasitizing the virus itself. But it's not really coding for anything genetically. So in a way, I think it's a sheep and a wolf skin, because from the particle exterior, it looks exactly like a virus, but it's sort of dead inside. It doesn't really carry any genetic information. It's not infectious. It just gets copied when the virus is already there. So these artificial sequences would have a replicative advantage over the wild type virus. They could be engineered to have an assembly advantage as well. So mathematically, it's quite clear that this therapeutic interfering particle would outcompete the actual virus, and that might be a good strategy to get rid of viral infections. So you can make some very nice molecular simulations, which we use Gillespie type algorithms for. So we keep track of things at molecular level. Some of you have seen a lot of SIR type modeling with COVID recently, but you can also look at molecules sort of in the cell, or how different parts of the immune system might interact with these viruses. And you could also make toy simulations of how assembly might work of such nanocages. So these are very high-powered, very computationally intensive molecular simulations. And yes, you could probably do a simple ODE model, but I liked Samir's quote from the talk the other day by Boltzmann. You might be missing some rather important things if you do the ODE approximation, because ultimately things are atomistic, as he says here. So just to show, in the cell, you can make some very complicated models of what's going on with the ribosomes making protein, with the polymerase making these virus sequences, but also these therapeutic interfering RNAs. They compete for polymerase, they compete for assembly, but typically the therapeutic interfering particles outcompete the native virus. And then you can look at it at the level of the immune system as well, see how viruses might infect cells, how that stimulates the immune system, and so on. You can make very elaborate simulations. But again, the bottom line is, of course, the tips outperform the native virus. So here, the pink line is these tips over the course of infection, co-infection with the virus, which is the blue one here, or as prophylaxis, as a vaccination. And both virus and tip grow exponentially. the tip is exponentially higher, and it essentially drives the virus to extinction in this prophylactic setup. You can also think of introducing it later on in a sort of chronic infection. And again, in this example, it drives the virus down to extinction quite quickly. So this might be a really interesting antivirus strategy going forward that exploits these insights into viral assembly. So you can also do these elaborate simulations for simulating how such a virus might assemble. So I've only got a couple of minutes left, but I wanted to talk about that very briefly. So you've probably all heard about the spherical cow as an approximation. So this is a dodecahedral cow. It's a toy model of a virus consisting of these 12 pentagons that we've already seen in the cartoons a few times. So you have these 12 packaging signals, 12 pentagons, and how well these packaging signals bind code protein and in which sequence determines how efficiently this particle can assemble. So there's a grading here, which is sort of weak, medium, and strong binding. And effectively, this gives you a 3 to the 12 dimensional fitness landscape that you can explore exactly and try to understand what the important principles are underlying virus assembly. So we've got these 3 bands of binding strength. We do very elaborate molecular simulations. This took 4 weeks on a supercomputer. And we give these sequences enough resources to make 2,000 capsids, and the measure is then how many of these 2,000 did they actually assemble successfully. So there's a fitness landscape similar to something you might have seen in John Hauenstein's talk the other day. So it's essentially a typical machine learning example. So this is work with Jan Hoyre that came out a couple of months ago. So you have these input vectors, which are 12-dimensional vectors. The output vectors are the scalars. And this black box was the expensive map, 4 weeks on a supercomputer doing these elaborate molecular dynamics simulations, which are computationally very costly. But we've heard of neural networks as sort of cheap approximations to maps because we can train the neural networks to become a good approximation. So Yang and I were looking at how well that worked when we were at Harvard and Brown together at ISERM a couple of years ago. And it's quite straightforward to map these 12 packaging signal vectors to the assembly fitness. And there's a lot of structure in that data that we could machine learn within a minute, what took this supercomputer 4 weeks. So the bottom line is there is some structure in this data set. It's not quite clear what that is. Some of it might be the ODE model approximation or the ODE approximation to this assembly model. There's also some other things like what seems to be good is having a definite starting point to the assembly. And then if all these binding strengths were really strong, actually what could happen is things misfold and get stuck and they don't assemble successfully. Whereas if you have some medium strengths here, it allows for some error correction. It's all thermodynamics, so things can bind, things can fall apart. So if you can make if you make a mistake, you can correct it if the binding isn't too strong. So that seems to be the type of lesson that we're learning by looking at what's successful. So trying to understand how we can couple machine learning usefully to simulations, trying to figure out what structure is in the data that might allow us to do more realistic examples that aren't just this dodecahedral toy model. And the other main point I would say is understanding these assembly instructions completely opens up new doors for antivirus strategies via these therapeutic interfering particles, which are really exciting, as well as some other applications in biomedicine and nanotechnology. So that was a bit of a whirlwind tour through structural virology. So there's more details on viral capsids and machine learning and these tips in the papers that I showed earlier. You know, I have other more algebraic interests, which I might talk about at the next incarnation of the Nankai Symposium. But just to finish on a bit of a plug, we're interested in machine learning and mathematical structures. So there's a topical collection that a few of the us people at the conference are co-editing. So Yang, Al Kaspchik, Andrew Lucas, and I. And this topical collection is called Machine Learning Mathematical Structures. It's in Advances in Applied Clifford Algebras. We know not everyone will do all three. Not everyone will do machine learning and mathematical structures and Clifford algebras. So some subset of these will be fine, or if you use that as a way to stimulate new collaborations, that's also something to think about. So if you have a machine learning mathematical structures paper that you're looking for a home for, do consider this topical collection. So thank you very much.
So I think we probably have 5 minutes for Pierre. Is there any questions? So, okay, so I have a quick question is that, so in the last part, I think you went quite fast with the machine learning part. So what do you actually envision machine can assist here, like to actually classify those models more efficiently than the previous supercomputers? Or
yeah, so in these models,
so what does this diagram really, like the one with a red blob there? So
that one, yeah, so it's plotting predicted versus actual.
Okay.
And it is quite a good fit. I don't think this is the best one you could do, but
Okay.
it shows that there's some inherent structure in this that could be learned. So if
So
you
the
use part of
individual
your data,
dots, I mean, yeah, sorry, what are the axes? So the, oh, I see. So the individual dots are the different models, those data. Okay.
So you get 500,000 data points where each of the 12 numbers here gives you the binding strength of one of these packaging signals. Okay. And these 12 binding strengths determine how well it assembles. So what this is finding is what the important aspects of this is. There's clearly a lot of degeneracy and redundancy and a lot of structure in the data. So we see already something that binds quite strongly at one end already has a very high fitness, even though there's only one very weak binding at the end. Having two strong binders at the beginning is already a very good thing. But this is only for a model that is nowhere near anything realistic, right? So this has 12 building blocks. Anything realistic might have 90 building blocks. So then looking at the molecular dynamic simulations you would have to do, it's just way beyond anything we can do to explore exhaustively the whole fitness space. But what we could do is explore different parts of the fitness space and then say, actually, machine learning was good enough last time. Maybe it's good enough this time, and we extrapolate. So that's the idea.
Okay,
so we can do more realistic things in combination with simulations and machine learning.
I see. Thanks. Any other questions?
Whilst I'm here, I'm just showing you the slide for what you asked earlier about these giant viruses.
Okay.
So there's various different types, and the lowest one has 147 as a triangulation number, and then various possibilities with different Ks. So all the H's are 7, Ks are 7, 8, 10, 12, 13, 18. And then the largest is 499. And the largest one that's known is called Mimi virus, and it will be something around 1,000, but the experimental resolution isn't good enough to really narrow down which triangulation it could be. So then you can look at the holes here. Does a particle with K equals 9 exist? Has it just not been found, or are there any reasons why maybe it's not favorable? But why do they all have H equals 7? Is that for making these little hooks and then it can sort of assemble? Is that the conserved feature? Because if you imagine they all have the same building block and they have the same little hook made by the H equals 7, all they need to do is be able to control that they can switch between having different Ks reasonably easily, and then they can all be evolutionarily related, even though they have vastly different numbers and sizes. So I can maybe show some of them. So as building blocks, they all seem to have a similar hexagonal shape and size, and they're the sort of fundamental tiling unit. And that probably doesn't change very much, because if you evolve away from that, it stops being able to assemble, and then obviously the virus dies out. So maybe that's heavily evolutionarily conserved. What can change, so the above is sort of a rotation by 90 degrees, and you're looking at this tiling unit in the plane, but you can have extra stuff coming out of the surface that doesn't have anything to do with the geometric tiling, but it's just a decoration on the surface. So sort of some fibrils or stuff coming out of the virus surface that they do disagree on, but not the fundamental tiling unit. That seems to obey the scaling behavior.
I see, thank you. So in view of time, I may have to cut Pierre short, but
yeah, no, that's fine.
So let's quickly thank him again. Yeah, thank you very much.
Thank you.
Very entertaining, actually.